Trillions of miles away are stars on the brink of death. Some dying stars end their life cycles with a brief but massive explosion of gas and radiation known as a supernova. Some supernovae are so energetic they emit a beam of intense radiation called a gamma-ray burst. Although many light years from us, astronomers think a gamma-ray burst could destroy the Earth's ozone layer. It's a remote chance, but its potential effects on life on Earth could be devastating.
So gamma-ray burst comes from a supernova. The gamma rays hit the atmosphere. Atmospheric chemists looking at this say that the gamma rays will interfere with the chemistry that makes ozone in such a way that the ozone would be eliminated.
If a gamma-ray burst destroyed our ozone layer, there would be nothing to protect us from invisible ultraviolet light from the sun.
So the sun feels very close to us, but actually we're being protected from the sun by the atmosphere that provides a barrier preventing invisible light like UV from getting down to life on Earth.
Ultraviolet, or UV light, is best known for causing sunburns, but it affects all aspects of the natural world.
So what would happen if the ozone layer disappeared? What would happen to life on Earth if there was a big increase in the UV coming on from the sun? That's the question that we're trying to answer.
Pat Neale is studying some of the smallest and most important organisms on Earth, phytoplankton.
These waters, like all waters on Earth, are full of microscopic plants. They're called phytoplankton, and they provide food for the rest of the marine food chain. They are the main source of photosynthesis in Earth's waters.
Ultraviolet light may disrupt photosynthesis within phytoplankton. If phytoplankton cannot photosynthesize, then they cannot create energy or food. Without phytoplankton, the rest of the marine food chain suffers.
We think aquatic organisms could be particularly vulnerable because they're very small and typically do not have structures that protect them very much from radiation.
Phytoplankton also store carbon. If the phytoplankton population decreases, there will be higher amounts of carbon dioxide in the atmosphere.
Phytoplankton are the main primary producers in aquatic systems, and the photosynthesis that's occurring in those systems accounts for about half of the biological sequestration of carbon on Earth. If that CO2 was not being absorbed, you'd see pretty immediate changes in carbon dioxide chemistry.
To study the effects of ultraviolet light on phytoplankton, Pat Neale and his colleagues have designed an instrument that simulates a world without an ozone layer.
The principle of our experiment is we try to simulate some of the UV changes that we expect would occur with big changes in ozone. We call our experimental setup the photoinhibitron. It looks at photosynthesis, but also looks at the inhibition of photosynthesis, so we have photoinhibitron. When we want to have fun around the lab, we call it the beast. So the heart of our system is a solar simulation lamp. It's just like the light from the sun, and as it's used here, it's like the light from the sun outside our atmosphere. So here's our sun, and here's our Earth, but there's no ozone in between.
Our cultures today?
Neale's team grows cultures of various species of phytoplankton. They put small samples of each culture into the photoinhibitron.
So the phytoplankton we're using in our experiments, we grow them in our pretend ocean. We take a little bit of seawater. We've added small amounts of different species of phytoplankton. These phytoplankton are going to go from their nice pretend ocean into the photoinhibitron, also known as the beast, where they'll be experiencing some not so nice conditions.
The photoinhibitron bombards the phytoplankton with destructive ultraviolet light, possibly making it harder for them to photosynthesize.
So the light is directed to this mirror. It's reflected off the mirror into exposure blocks. Each block is covered by a filter. Each filter blocks a different part of the UV spectrum.
Each sample in the photoinhibitron is exposed to a different wavelength of UV light, starting with the shorter wavelengths, all the way up to the longer UV wavelengths that would occur if a gamma ray burst destroyed the entire ozone.
And then we trace how much of that carbon is assimilated with time. When the carbon is taken from a mineral form and it's incorporated into the cells of the phytoplankton, that's the photosynthetic process.
Here we have our samples that were exposed yesterday to our photoinhibitron. So we have, you can see 120 sample vials here, and they get counted on the scintillation counter. So we can actually count the amount of organic carbon that has been incorporated to determine how much photosynthesis each cell has done.
A machine called the scintillation counter measures how much of the inorganic carbon has become organic as a result of photosynthesis. How much the phytoplankton photosynthesize indicates how much they have been affected by the ultraviolet light.
This is our scintillation counter. We count how much of the carbon has been incorporated in that hour exposure. So for the high UV treatments, less photosynthesis will happen, so less carbon has been incorporated. For the low UV ambient conditions, higher carbon has been incorporated, more photosynthesis.
The initial results for one species of phytoplankton exposed to UV radiation show a definite reduction in photosynthetic activity, but not enough to have a major effect on the rest of the marine life food chain.
So the more UV that they get, the less photosynthesis. That's the basic thing. There's so far only small additional effects of the extra UV with a gamma ray burst. And it's probably not enough to make the difference between extinction or survival of the organisms that are dependent on it. It is enough that would have some significant effects on productivity. That is just what we've seen so far for one of the species in the ocean.
But Neale is also testing other smaller and more abundant types of phytoplankton that might be more vulnerable to ultraviolet light.
So we've only seen modest responses in one species, but we're looking at other species, and we do have some indications that other species are much more sensitive to UV exposure than this Synechococcus species that we have been looking at.
More data will help reveal how life will be affected if the Earth's atmosphere is exposed to a gamma ray burst.